Hello, welcome to the Knowledge Catalog. This is CJ, and I hope that you're not that you like our new look. Okay, this is going to be the new look, the new format of our succeeding videos. If you notice that in this video we are featuring learning competency, familiar yan siguro sa mga public school teachers natin dyan. Hello. Hi. This is this is a video which we may use in Science 8. Okay, so in a specific learning competency called compare mitosis and meiosis and their role in the cell division cycle. This is the fourth learning competency for that quarter. If you want to have a copy of this PowerPoint presentation, you may contact me, send me a message on facebook.com /knowledge catalog /. Nilagay ko talaga yung / yan kasi parang feeling ko kailangan siya. So yeah, let's begin. Okay, so in this video we're going to talk about the stages of cell division, or the stages of the cell cycle. Actually, they're not the same, and this is why they are not. This circle represents the entirety of cell cycle. As you can see, this entire circle is divided into number 1, G1 phase, number 2 is S phase, and then third is G2 phase, and then this one is the mitotic phase, or in the case of meiosis, it's the meiotic phase, if that's even a word. Okay, meiotic. This entire circle is the cell cycle. Itong meiotic phase naman is the actual cell division phase. Diyan talaga nagde-divide yung cell. Okay, all of the processes that lead to the division of the cell are actually here in the entire cell cycle, but the actual division of the cell is in the mitotic or meiotic phase. Now, interphase is comprised of the following parts or stages. The first one is the G1 phase, and that is what we'll be discussing here. The next part of interphase is the S phase, and then the third part is the G2 phase. And then interphase is going to end with G2, and then mitotic phase is going to take place. It's called the cycle anyway kasi after the meiotic phase, yung mga daughter cells, pwede rin silang mag-continue to G1 phase again para tuloy-tuloy yung pagde-divide. Okay, so that is if ready na ulit siya na mag-divide. Okay, now, we start the discussion with the interphase, the first part, which is G1 phase. For G1 phase, here, we take note of the words cell growth. The cell duplicates its organelles, and then it also grows in size. Because the cell is duplicating its organelles, syempre, ang magiging resulta nun is mangangailangan na mas maraming space. Okay, the cell cannot stay on that particular size, the size na meron siya before nung isa lang yung mga, or I mean, yung meron lang siyang enough number of organelles for it to survive. Kasi ngayon, dinuplicate niya ng mga yon. So, given lang talaga na lalaki na yung cell. Because, yun nga, dumami ng organelles niya. Another one is that it builds the molecular building blocks in the later steps. All of the molecules, all of the macromolecules that are needed for the next steps are being built, are being produced na as early as G1 phase. Ayun. So, remember, that is the first step in interphase. So the next step is what we call as S phase. Now, looking back at this illustration, S phase, meron siyang process dito, ang tawag sa kanya is DNA synthesis. In biology, when we say lysis, it is the breaking down of things. When we say synthesis naman, this is the use of different molecules to build something up, to produce something, okay? Like combining these molecules together, that's quite redundant, for you to be able to produce a new product, which is, the process is called synthesizing. Okay, so I hope that makes sense. For the synthesis phase kasi, ang nangyayari dyan is, yung DNA, sa loob ng nucleus, okay? Remember that the DNA is found inside the nucleus. Is being duplicated. Okay? Again, the DNA of the cell that is found inside its nucleus, it's duplicated. Diba? Lahat naman ng mga pati yung organelles na duplicate. Even now for synthesis phase, nakaduplicate na rin. Dinoduplicate na rin yung DNA. Now, aside from that, dinoduplicate na rin yung mga centrosomes. If you're familiar with the word centrosome, then that's okay. It's an organelle that produces the microtubules, yung mga spindle fibers na later on magiging talagang napaka-importante for the actual cell division. Okay? Next one is the G2 phase. G2 phase comes after S phase, ayan, and it also entails here cell growth, just like G1. For the G2 phase, the following are the things that we need to remember. So the cell continues growing, and then it makes more proteins, and then it makes more organelles. Okay, so it's more like ganito, because the cell is producing more proteins and duplicating more organelles, it is growing. Okay, that is a more appropriate way of saying it. Another one is that it reorganizes its contents in preparation for prophase or prophase I in case of meiosis. Nire-reorganize na niya yung content niya kasi papunta na niyan siya talaga ng actual cell division phase. Now, mitosis is a form of cell division. It's one of the two M's na i-discuss natin in this video. The second one is meiosis. Let's first dwell on mitosis. Mitosis, it happens among somatic or body cells. Again, mitosis happens among somatic or body cells. Now, let us first study this illustration na kinuha natin over the internet. Okay. So interphase, G2 phase, ayan na siya. It's going to proceed with prophase. Okay. Prometaphase, which is a stage between prophase and metaphase, so it's easy to remember. Metaphase, and then anaphase, and the last one is telophase and cytokinesis. Some references are saying that cytokinesis is happening after telophase. Some references naman are saying that cytokinesis is one of the events that are happening during telophase. So either way, madali lang, it's siguro for basic education, it works that we say this phase is telophase, and then one of the events that are happening in it is cytokinesis. Okay, at least for the purpose of discussing it with the kids. Ayan, so prophase is, as we see, it's the first stage during mitosis. Ayan siya. So, this is the nuclear envelope, and then these are the centrosomes. Ayan, meron silang yellow fibers. Prometaphase naman, yung mga yellow fibers are scattered, tapos wala na rin yung nuclear envelope, nakalabas na rin sila. Ayan. Tapos yung mga parang tali-tali strings lang na yan, naging mga chromosomes na sila dito by this stage. Yes. So, the centrosomes are found on the opposite sides. Yeah. And then for the metaphase naman, naka-line up na sila sa may bandang gitna. Okay, that is a good lining up though. Ayan, yung centrosomes nasa may magkabilang dulo tapos parang they seem to meet na talaga ngayon. Right? And then for the anaphase naman, pinaghiwalay na yung mga sister chromatids, they're separated, they are being pulled by the centrosomes on the opposite sides. Tapos pansin nyo rin itong fiber na to ng this is spindle fiber, nakatuloy-tuloy siya, di ba? Bakit kaya? And then the last one is meron na siyang ayan, parang okay, basta parang namumuo na yung division. Ayaw mo na sabihin yung word. Diyan sa gitna ng dalawang parang daughter cells. Ayan. Okay, so again, remember this is happening among somatic cells. Okay, now let's dig deeper. Mitosis adds new cells during the periods of development. Yung mga days or yung mga months na produce mo na pa tayo ng mga organs or organogenesis, or yung pag-grow natin in size, that is yung pagbe-build natin ng bones, pagbe-build natin ng bagong muscles, yung pag-repair, ayan, lahat ng mga yon during those periods of development. Mitosis is adding new cells. Okay, just as in this case, from one cell, meron ng dalawang daughter cells. Now, it also allows the replacement of worn-out cells throughout our lifetime. One good example of this is, skin. We are shedding skin. We don't have the same, the same skin na meron tayo before. Okay, otherwise, just come to think of it, paano nagkasya yung mga buto mo, yung mga laman mo dun sa skin na yun, di ba? Parang para sa mga malinta. Just common sense lang siguro yun. Kaya na nating isipin yun. That, since cells are dividing, our bodies are also bound to, our bodies are bound to grow. Okay, since one of the signals that we need for cell division anyway is mitogen, or yung mga growth factors, right? So, yeah, I hope that makes sense. Another one is its goal is to produce daughter cells that are like the parent cell. Kailangan ang mga daughter cells na to kamukha siya ng parent cell. By saying kamukha, we are actually more particular with the number of chromosomes that are found sa may daughter cells at sa may parent cell. Dapat magkapareho sila ng number of chromosomes. Okay, so the first stage in mitosis is called prophase. So let's just study muna this illustration. This illustration came from Khan Academy. I love their illustrations. So early prophase, we have the following. Mitotic spindle starts to form. Ayan siya. Yung pinakita rin natin kanina dun sa unang illustration. Centrosomes ang tawag sa mga to. Tapos spindle, mitotic spindle fibers. Ayan sila. Then the chromosomes are starting to condense. And then the nucleus is gone. Ang nangyayari dyan is yung nuclear envelope actually ang nawawala. Yung covering, yung membrane na bumabalot sa nucleus ang nawawala. Nucleolus pala, sorry. Yung nucleolus ang nawala. Okay. Yung membrane, or actually walang membrane yun. Walang membrane ang nucleolus. Basta it's just a place near the chromatids na responsible sa pagproduce ng ribosomes. Okay. So, some structures break down while some others naman are being built up during the early prophase. The mitotic spindle forms, and then they are actually microtubules that move chromosomes around during mitosis. They grow between centrosomes. It refers to these fibers. Okay, it refers to these fibers. Ang sabi sa kanila is sila yung mag-move around, sila yung maggagalaw ng mga chromosomes during mitosis. Okay. And they are also found between the centrosomes. Okay. So next one is the nucleolus disappears. Yun nga ang area. Okay, let's be clear. A nucleolus is not an organelle. Again, a nucleolus is not an organelle. What is it then? It is a place near the sister chromatids inside the nucleus, and those sister chromatids are responsible for giving the instruction on producing ribosomes. Okay, I hope that is clear. Now, the nucleolus. It is the organelle responsible for the production of ribosomes. I am correcting this. Again, it is not an organelle. It is just an area near the chromatids inside. Of course, down the comments, I am open for more ideas because my research is just based off from around 2 to 3 literature. So I hope that you would be able to help me in understanding or having more information about this. But for the meantime, yung marami sa mga references ko are saying that nucleolus is not an organelle. So late prophase now which is called Prometaphase. Let's observe. As you can see, the nuclear envelope is already broken down, and then the chromosomes are fully condensed. They thickened. From being stringy, they became now as sister chromatids bound together sa may centromere. The mitotic spindle attaches now to the chromosomes. So, nag-start na silang mag-catch ng mga chromosomes. As the chromosomes continue to condense, and the nuclear membrane breaks down, which releases the chromosomes. Remember na yung nuclear membrane encloses the nucleus, and inside the nucleus, in that place is are the chromosomes suspended in the nucleoplasm. Okay. Now, the next part of prophase now is this. Let's have a closer look at the spindle anatomy. These are the centrosomes. These are the spindle fibers. Ayan, spindle fiber ang tawag dyan. Spindle fibers. There. Okay. Now, a spindle fiber is a microtubule. Again, a spindle fiber is a microtubule. If that spindle fiber or that microtubule is attached to the centromere, here, attached to the centromere, or near the centromere to the kinetochore, you call that microtubule as a kinetochore microtubule. Again, an area near the centromere, we find these kinetochore, areas near the centromere. If the microtubule or the spindle fiber is attached to it, you call that a kinetochore microtubule. Okay? And those kinetochore microtubules are also spindle fibers. Okay? It's just that they are connected to the kinetochore. Now, you see that there's also this word called aster. Okay, so sila yung mga sobrang kasi nag-elongate pa rin yung mga nag-elongate pa rin yung spindle fiber sa may sa may opposite side nung centrosome, and you call that as aster. Now let's talk about the details. The microtubules attach to the kinetochore, which is a patch of protein found in the centromere. Since it's a protein, it is specifically serving that purpose. Now, once the microtubules are attached to the kinetochore, they will be called kinetochore microtubules. Okay, so umulit lang tayo doon. In case a microtubule did not attach to a kinetochore, it reaches a microtubule from the opposite side, just like this one. This microtubule, it did not touch any kinetochore, therefore, it reached out to the opposite, to the microtubule on the opposite side. Ayan, nag-meet sila, and the purpose of that is to make the spindle more stable. Ayun. After the okay, so now we're done with the prophase. We're done with the early prophase, and then we're done with the late prophase called prometaphase. Okay, so by now, question, ano nang nangyari ngayon? Anong nangyari ulit sa sa prophase? Okay, so nag-attach na yung mga spindle fibers sa kinetochore. Tapos, 'di ba, ang gagawin ng mga spindle fibers, sila yung maggagalaw ng mga chromosomes. So, ang gagawin nila niyan, gagalawin nga nila yung mga chromosomes. Saan nila ilalagay? Paano yung paggalaw? Ilalagay nila sa may bandang central plate or the metaphase plate. Take note that the metaphase plate is not an actual thing. Okay? It is just a term that we use to refer to the place kung saan banda naka-line up yung mga chromosomes. So, chromosomes are lining up at the metaphase plate. Again, the metaphase plate is not an actual part of the cell. It is just really the term we use to refer to the lining up of the mga chromosomes. Now, the spindle has captured all of the chromosomes, and they are lined up at the metaphase plate. You have to take note that, again, metaphase plate is not a physical structure. Comes next is anaphase. For anaphase, let's observe the following illustration. Okay, so anaphase, from the word and let's take note of the word ana, which means breaking down, if I'm not mistaken. Kasi pag analyze, it also means double breaking down. Kasi lyse means breaking down, ana, which means breaking down. So in anaphase, ayun, pagse-separate na yung mga sister chromatids. Yung kinetochore microtubules are going to pull the chromosomes towards the poles. The microtubules push poles apart. Okay, so ano ulit yung two types of microtubules na sinasabi dito? Yung mga microtubules na walang pinag-attach na kinetochore at yung mga microtubules naman na mayroong pinag-attach na kinetochore. For the microtubules that attach to kinetochores, they will be pulling the chromosomes towards the poles where they belong. For microtubules naman that did not connect to a kinetochore, attached to a kinetochore, they will be pushing the poles apart. Okay, so totally separating the components of the later on daughter cells. So the glue that attaches the sister chromatids together is broken, and it allowed them to separate. The microtubules are not attached to the chromosomes. The microtubules that are not attached to the chromosomes elongate and push apart, making the cell appear longer, just like in this illustration. Okay, so in Anaphase, remember that what happened there is pinaghiwalay na yung mga sister chromatids, yung mga chromosomes. Now let's observe Telophase. Telophase, telo is ending. So chromosomes start to decondense. So naglulusen up na ulit sila. Okay, naging stringy na ulit sila. And then yung mga spindle naman, spindle fibers they disappeared. The nuclear membrane is starting to form again, and then the nucleolus is again reappearing. Remember that since meron na nga may mga chromatids na nga dito, okay, the reason why nagre-reappear ng nucleolus is because meron ng mga chromatids dyan. Okay, so telophase. The mitotic spindle is broken down into its building blocks, yung component molecules niya. The two nuclei, nucleoli, and nuclear membranes form, and then chromosomes decondense to return to their springy, stringy form. Ayan, ganyan yung form nila. So in the telophase also, we take note of the word cytokinesis, or the movement of the cell. Cell kinesis is movement. So among animal cells, ang nangyayari is masyadong contractile yung cytokinesis nila. There is this deformation in the parent cell. What appeared to be a parent cell before, there is this deformation called cleavage furrow. The actin ring is also here. It is actin is a protein, more exactly an enzyme if I'm not mistaken. Now, ang ginagawa niya is just like a sling bag. Ginagawa niya is pinagse-separate niya yung dalawang daughter cells from each other. Kumbaga, hinigpitan niya yung area na yan hanggang sa maputol yung connection. Okay? So, yun namang part na to is what you call as the cleavage furrow. And remember, this happens among animal cells. Okay? Among plant cells naman, hindi naman sila capable of doing this, yung mga plant cells. Ang nangyayari na sa kanila is cell plate is going to form between the two daughter cells. And that cell plate is found inside a cell wall, which is later on going to connect to that cell plate. Ayan, to finally divide them into two. So next detail is that we are now going to move to meiosis. So remember that telophase and cytokinesis ends up mitosis. So now we have here daughter cells that are haploid. Let us not use that word yet. Okay, so kailangan meron tayong separate na discussion about that. So let's just say that the daughter cells have the same characteristics as the parent cell. So later nang natin pag-usapan kung haploid ba talaga yan or diploid. Okay, so for the sake of our furtherly discussing what we mean about haploid and diploid. Next time lang yan. So now, we talk about meiosis. Meiosis is a form of cell division that happens among gonadal or sex cells. So, among humans, sa mga egg cells, sa mga sperm cells. So, let's observe first the illustration. Okay, so, kung kanina, meron lang tayong ilang mga circles na na-observe dito, marami-rami. Yeah. And then for Prophase I, ito yung buong yan. Prophase I, Metaphase I, Anaphase I, Telophase I, and cytokinesis. And then Prophase II, Metaphase II, Anaphase II, and Telophase II, and cytokinesis. So, what do we observe here? Prophase, meron tayong mga, okay, parang kakaiba to, no? Because we have here a red and blue, na parang chromosomes na yung blue kaso parang pinag-pair pa siya dito sa red. Pinair pa siya sa red. Tama? Okay, tingnan nyo. Yan. And then look at Anaphase I. Parang teddy bears yung pinagse-separate, di ba? Meron silang 1, 2, 3, 4. 1, 2, 3, 4 na mga ano. Whereas kanina, di ba, hindi naman ganyan itsura niyan. And then for Telophase, as you can see, chromosomes sila. Okay. Now for Telophase I, those same chromosomes. And then Prophase II, Metaphase II, you see them, chromosomes. And then Anaphase II, ayan. So ito kahawig na nung sa Mitosis kanina. And then Telophase and cytokinesis. So now let's deal with the details. The goal of meiosis is the production of sex cells, that is to say, sperm cells and egg cells, that has half the number of the chromosomes from the parent cell. Question: if the daughter cells here has half the number of the chromosomes from the parent cell, does that mean that the parent cell and the daughter cells are alike? Yes or no? Of course, the answer is no. They are not alike. Okay, so let's begin with Prophase I. For Prophase I, we see the following illustration, contrary to the circle na ginagamit natin kanina sa mitosis. So, meron tayong ganito. So, observe that for the first pair, ito, first pair, second pair, third pair. They're actually the same chromosomes, okay? It's just that ang pinapakita dito is nangyari sa kanila is naging ganito sila, tapos naging ganito sila. Okay, so now let's begin. This is an entirely red chromosome with uppercase letters. So this is A, B, C of this chromatid. This is A, B, C of this chromatid. For this naman, purplish ba to? Bluish for you. Okay, so whatever. It uses lowercase letters. You have lowercase a, b, and c for this for this chromatid, and it uses lowercase a, b, and c for this chromatid. Now, what happens to them is they will be they sort of like paired up. Parang they're touching each other now. Okay, so A, B, and C. So ito A, B, and C uppercase. Tama? And then this is lowercase a, b, and c dito. And then A, B, C. Now, what happens next is, charan, ayan. So, nag-separate ulit sila. What happens here is A, B, C, same from the original. Whereas this one is A, B, and di man halata pero lowercase c. And this is color blue or purple. I mean violet. Violet, blue. Tama? Pareho ba yung perception natin ng color? Basta lowercase. Okay? Parang nahawa siya yata dito. Parang sa labada, nahawa siya dahil nagkalapit sila, no? And then this one naman is A, B, and then uppercase C. So, they're no longer the same as the original. Okay. So, what happened in Prophase I? Exactly. So, chromosomes condense, just as, such as this one, nag-condense sila, and they pair up. Okay, so remember the term pair up. With their homologous partners. They match up, aligning according to their length. So, ito pala, si chromosome, red chromosome, si blue chromosome, hindi ko talaga sure kung ano bang kulay nito. So, si red chromosome and blue chromosome are homologous kasi they have the same length and their sites are fitting. Ayan, so A, B, C, C, N. So they're homologous. And then they're going to pair up. So yun pala yung term. They paired up. Nagpares sila. Now with these pairings, crossing over happens. Now what is crossing over? The chromosomes will be on top of the other, bound by a protein called synaptonymal complex. They're actually on top of the another one. Okay, pinakita lang talaga dito is para makita nyo na magkasama sila. Kasi pag pinagpatong lang ang dalawang yan in an illustration, baka hindi mahalata. Okay, but in this explanation, nakapatong talaga sila dyan. Now, crossing over is the process of the sharing of the traits na nangyayari. In this case, isang trait lang ang na-transfer sa isang chromosome, okay, sa magkabilang parts niyan, ayan. Isang trait lang, yung C, for this chromatid. So, the synaptonymal complex is binding them in such a way that they look like this. So, ang homologous chromosomes dito is yung red at saka si blue, red and blue. And then they are sister chromatids, and then yung dalawang blue are sister chromatids. Now, continuation of Prophase I is that let's first deal with the illustration. So what do you see? For A, B, and C. So in A, they are homologous pairs. Okay, pareho yung length, tama ba? And itong isang to, ayan, at saka itong isang to are sister chromatids. Okay, this is a chromosome, and then this is also a chromosome. They are homologous because their lengths are fitting. And then second one is the chiasma is formed. Ano kaya yung chiasma? Obviously, it is the region where the two homologous chromosomes are connected. Okay, that's more appropriate. The chiasma is the region where the homologous chromosomes are connected or nag-cross over. And then C, obviously, yung tulad ng pinakita natin example kanina, itong si blue chromosome, naroon na siyang red because nakipag-crossover siya sa ka-pair niya. Ayan. So, even red has now a blue, a color blue from the blue chromosome na naka-pair niya. Now, through a microscope, crossing overs are viewed as chiasmata. These lengths keep the homologs together after the breaking down of the synaptonymal complex. Yung kaninang pinakita natin na synaptonymal complex, this one, mabibreak down yan. Okay, mawawala rin yung force na nagbabind sa dalawang chromosomes na to. And what is going to remain is the chiasma. Siya lang yung matitira na magbabind sa dalawang chromosomes. Ayan. Now, these crossing overs are random. Napaka-random na mga yan. In nature, pwedeng meron pang... Actually, hindi lang sila mag-crossing over sa isang part lang tulad nito at nung previous example. Pwede actually marami yung mga pagpapalitan nila ng mga traits. And they are completely random. And because of that randomness, nagiging unique yung traits ng isang... yung combination ng mga alleles. Okay? Yung traits din nami-mix up. Kaya unique na unique yung magiging products dito. Yung mga daughter cells are very unique. Okay, so Metaphase I, tulad nung kanina, meron pa ring central plate. Okay, so spindles, by now, I hope you remember what spindles are. They are these guys. Capture the chromosomes and line them up. Okay, line up the homologous pairs at the central plate with a random orientation. Itong mga homologous pairs, they line up at the metaphase plate, and what do you call them? I remember yata, in my college, during college, ang tawag namin dito yata is tetrads. Yeah, tetrads. 1, 2, 3, 4. Tetrads. Alright, note, each chromosome attaches to microtubules from one pole only. Okay, now, this is quite the work. So, focus on the illustration. This spindle is going to connect to one centromere. Ito yung kanina, di ba? One area that is near to the centromere. Kanina, it's the kinetochore. However, ang mangyayari ngayon is that it's only going to connect to one of the two chromosomes. Okay? So, if that is so, di ba, we know na itong mga kinetochore microtubules, they will be pulling them. And then itong namang mga spindle, nitong mga free microtubules naman, they are going to push each other apart para mag-elongate yung cell. So that means it's going to separate these two homologous pairs from each other. Okay? So paghiwalayan is yung homologous pairs, hindi yung mga sister chromatids. Such as so, and as so like so. Okay, so this is Anaphase I. Homologs separate to opposite ends of the cell as shown, while the sister chromatids stay together as shown. Okay, so homologs are pulled apart to the opposite poles. Upon separation, the sister chromatids remain together and they do not come apart. This is very different from mitosis, right? Okay, iba yung anaphase na mitosis dito sa meiosis anaphase I. And then next is telophase. So the newly forming cells are haploid, which is n is equal to 2. Okay, so each chromosome has two non-identical sister chromatids. So yung mga to, they are not identical to this one. Yung daughter cells, they are not identical. But are we done with meiosis? Of course not. After the cytokinesis occurs, okay, there will be two daughter cells that are haploid. Again, we'll be having a separate discussion about that. In some organisms, the chromosomes decondense and the nuclear envelope reforms. But in some, these steps are skipped since the daughters will soon undergo another series of division called Meiosis II. Meiosis II has the first step, Prophase 2, which is obvious. Alright, so it starts with cells which are haploid that are made from Meiosis I. So chromosomes are going to condense, familiar na to, no? While the nuclear envelope breaks down, familiar. The microtubules are released from the centrosomes, beginning the capture of chromosomes. Metaphase 2, familiar din? Since nakapag-capture na sila ang mga spindle fibers ng mga chromosomes, ilalagay na nila yung mga yon sa central plate. And then some of the spindle fibers are free, some of them are kinetochore microtubules. Okay, so each of the sister chromatids are captured by microtubules from opposite poles. Now, the microtubules are going to line up at the chromosomes in the central plate. Again, hindi naman basta-basta nagpunta dyan ang mga chromosomes like trip lang nila. It's not like that, okay? They are being controlled or manipulated by the spindle fibers. Now, for anaphase, this looks like mitosis now. The sister chromatids separate to opposite ends of the cell. Since they are being pulled to the opposite poles of the cell by these kinetochore microtubules, the free microtubules now are going to push naman each other apart para mag-elongate yung buong cell. Now, the sister chromatids are pulled apart and they are separating the sister chromatids. So, the nuclear membrane reforms, mawawala na rin yung mga spindle, and the chromosomes are going to decondense. So, ayan, meron pa rin cytokinesis na nangyayari dyan. Okay, so may cleavage furrow? Yes. Okay, so I hope that this video helped us in understanding the steps of, cell division or the cell cycle as a whole. I just would like to clarify that this is providing us much like an overview. Kasi ang alam ko, meron pa mga deeper pang mga processes dyan. At saka marami sila talagang mga enzymes na nagmamanage ng mga reactions at ng mga activities in the cell. For this video, I would like you to remember that this is sort of an overview para rin sa mga beginners ng molecular biology. A separate video is going to be produced for explaining haploid, diploid, those terms. Kasi siguro we need to talk more about genetics first before we use those terms with frequency in these videos. So the Knowledge Catalog is a brand being developed by yours truly, who is a teacher creator. I am a high school teacher in the Philippines, particularly in the division of Angeles City, and in my school, Clara M. Recto, Information and Communication Technology High School. And I love developing content that I find useful and that are needed for my instruction. I hope that you will be supporting me so that I will be more motivated, I will be more motivated. I'm already motivated, but I will be more motivated if I see that you are enjoying these content that I am producing. If you have some criticisms, please channel them properly. You can always message me in my page and I will gladly hear out from you. I'm syempre a beginning creator, so I hope that you will be kinder when it comes to your critical comments. And if you appreciated my work, you can also leave a like or perhaps subscribe to this channel. I would like to, as much as possible, leave a legacy by producing as much as possible mga useful na mga content for us, because I'm also producing these for my own use. Okay, so this is the Knowledge Catalog, Education Solutions. In my time, good evening.